Okay, we're going to start the next session. And this section is on advances in disease predictors and the influence of the microbiome. And different people have different perspectives. So I'm going to first introduce them, and then I'm going to start asking them some questions. And we should have a little bit of time for the audience to ask questions as well. So Sónia, why don't you start, and we'll come this way, but for introductions, just introduce yourself.
Yes. Okay, hi everyone. So my name is Sónia Gonçalves Pereira. I come from Portugal Polytechnic Institute of Leiria. I'm a microbiologist, and I'm also a physician. In microbiology, I started working in celiac disease microbiome recently, about 3 years ago. So that's that.
Hello, my name is Rok Seon Choung. I'm from Mayo Clinic. I'm working with Dr. Morey, and Dr. Morey is really expert in celiac world. And also, my main focus is actually epidemiology and clinical stuff. I'm trained gastroenterology, but in South Korea, and doing research mostly in the United States right now.
Hi, my name's Michael Fitzpatrick. I'm an adult gastroenterologist focusing on celiac disease, but also aspects of clinical nutrition. And I also run a research group which focuses mainly on celiac disease using techniques like spatial transcriptomics and also these biomarker studies that we've been doing.
Hi, I'm Shalev Itzkovitz. I'm from the Weizmann Institute. I'm a systems biologist. In my lab, we're interested in understanding how cells work together in organs in our body. So we're going to use techniques like single-cell transcriptomics and spatial transcriptomics. We're very excited about the gut, the liver, the pancreas, metabolic organs. Specifically, the gut has this beautiful layered organization of cells along the villus axis, and that got me excited about celiac, where this entire morphology collapses. And we were very curious to understand how that impacts the physiology of the gut. So that's my connection to celiac.
Thank you. So we sometimes hear the term bench to bedside. So usually it means doing, when we say bench, more, I would say, oftentimes non-human work. But it can be human, but at the bench and not in a patient, to bedside, meaning the transition from learning things in a laboratory to getting it to a patient. So for each of you, I'd like you to talk about the research you're doing and how you think it will become applicable to either treatment or to diagnosis. So why don't we start with Sónia, if you could talk a little bit about your research and how you see that going from the bench to the bedside.
Yeah. Okay, so as a microbiologist, my main focus, it's two. So first, I'm interested in characterizing the gut microbiome of celiac disease and non-celiac first-degree family-related person, non-celiac. And with that in mind, I'm looking for differences that can guide possible biological treatments, probiotics, fecal transplantation, etc. So there's this huge variability of options. So I'm a very bench-located research. So before going, not the bedside, because fortunately the patients are not in a hospital. So I'm not celiac, but celiac patients have a regular life, etc. But I think my idea, my final goal at the end in this characterization of gut microbiome would be to deliver some information that can guide probiotics treatment. So Jocelyn said, so it's not gluten-free diet, it's not the treatment, it's a treatment. So maybe we can add up a probiotic or something that can modulate the gut microbiome and reduce symptoms. So that's one part. And so I'm here for this gut microbiome part of my research, but I also have some culture-based research, which is starting now. I isolated bacteria from celiac patients and non-celiac people to see how bacteria can digest gliadin, if they digest it, if they can contribute to it more or less immunogenic epitopes for the immune system, and if they can contribute more or less with inflammation and with translocation. So hopefully in the future, this very preliminary work can contribute to guide future research in mechanisms associated with the disease that at the end may lead to some kind of drug to not contradict, I'm not getting the correct word, but to prevent this inflammatory, this translocation, this immune activation of the disease. So that's that, being in the bench, but aiming for the bedside perspective in the long run.
Thank you. Roxanne?
I'm a trained gastroenterologist. I know about actually what kind of disease we have in terms of in gastroenterology and area. Because my actually main interest part is epidemiology. Epidemiology is meaning how frequent some disease actually occur in the population. And then how frequently, what kind of causal effect, causes can develop or even trigger and augmenting the disease development. Recent my research is focusing about the celiac disease. And as you know, the celiac disease is recently abruptly increased, but we don't know why. And what kind of factors actually involved? Is there an environmental factor involved? Or is it genetic factors involved? Genetic factors, we think, maybe very stable, maybe little tweaking, but lots of environmental factors involved that. And I'm from South Korea. We have very big epidemiology events in colon cancer and IBD actually. So about 20 years ago, I see the patient about 1 out of 100 maybe having IBD at the time. But about 10 years ago, I came back and I found about more than 10 out of 100,000 people actually having IBD. That's also happened really abruptly happening about increasing evidence of this IBD. And celiac disease, our group is really focusing about what really triggering the celiac disease and how can we find it early enough and can we alter the disease progression, especially from autoimmunity and to actual developing clinically obvious disease. I think it's very good example is type 1 diabetics. I think type 1 diabetics, the diagnostic criteria of type 1 diabetics quite changed. They're gonna usually right now is much more relying on the antibodies against the self-antibodies. Right now, the finding some antibodies for diagnosis of type 1 diabetics. So that's meaning with celiac disease also, it could find from early enough. And like in our study and our research area, we can looking about some environmental factors like exposome data or proteomics. You can and changes of proteome, evolution of proteins, and also evolution of immunology recognition, which is meaning some people may have already developed gliadin recognition initially. Maybe some people may recognize of TTG first, or then maybe gliadin TTG altogether. And those things could happen even before after having tissue disruptions. And we found about some of them actually have positive TTG IgA, which is meaning they may have celiac disease. But later, about 5 years later, even though they are on gluten-containing diet, we found they are actually regressed, which meaning the negative celiac disease serology testing. So we really want to know about what markers can predicting this disease progression. That's my main area. From even funding from Beyond Celiac is actually targeting that one. Hopefully, based on our research, and we can deliver really great markers for predicting disease, the progression.
Thank you.
So in Oxford, the unit that I work in is called the Translational Gastroenterology and Liver Unit. And the translational bit is all about this bench to bedside and back again and learning from each other. So we are a group of scientists, clinicians, and people like me who sort of straddle the fence a bit, working on a variety of gastrointestinal and liver disorders. So in our celiac group, we have some projects that are very translational. So our biomarker project and work has a specific clinical goal. Can we find potential tools or markers that could be used as a test to monitor celiac disease? So that has a direct translational benefit. But some of our other work, so we have a project that's looking at a technique called spatial transcriptomics, which is looking at where genes are expressed in tissue, what cells are expressing them, and what cells are talking to whom in the tissue in celiac disease. And that more fundamental research often leads to unexpected findings. And I think it's important to acknowledge that fundamental research often, in a surprising way, ends up leading to really important translational breakthroughs, but often in an unpredictable way. So you can be looking for one thing, but you find something else that looks curious or interesting, and that ends up being the target for a new drug or a new test or something like that. So I think in our unit, one of the strengths of the work that we do is because we have a good mix of people from a clinical and sort of fundamental science background, we can ideas can kind of go both ways. So ideas from maybe more kind of clinical research can inspire more fundamental science questions, and then vice versa, things can come through to the clinic.
Thank you.
In order to understand human diseases, we need to measure cells, to measure all of the molecules in cells from the affected tissues. And in terms of celiac disease, that involves, of course, taking biopsies, but taking biopsies requires an invasive method, and also we don't have access to the entire gut. We have access to certain parts of it. And so there's an alternative approach in medicine in general that's very exciting in recent years, and that's the approach of the liquid biopsy. The idea is that cells die in the body all the time, and they're shed, and we can find them, for example, in the bloodstream. If we take a blood test, we can measure RNA or DNA molecules, and we can diagnose certain diseases. We can find a cancer somewhere in the body. In the gastrointestinal tract, this is a little bit less relevant because cells are shed all the time, but they're not shed into the bloodstream. They're actually shed into the outside cavities, into the lumen of the gut. And what we hope to do is to measure deeply and entirely all of the cells that are shed into the lumen from stool samples. And I think this has a potential to both monitor disease of the gastrointestinal tract with a high temporal resolution in a non-invasive manner, and also personalize treatment. So, for example, in inflammatory bowel diseases, you can find specific signals that would instruct which types of biological drugs would fit the patient. In celiac disease, again, it could be that different patients could benefit from different kinds of drugs. We can also use these kind of sort of stool liquid biopsies to monitor functionally how the tissue regenerates after a certain drug or after a gluten-free diet. So I'm very excited about this approach of basically measuring dozens and hopefully hundreds of stool samples at the molecular level. And I think that even in a sort of a machine learning non-biased approach, that would give us a lot of information, subclasses of diseases, and this is something that excites me in the lab.
So we just had a session about clinical trials, and some of it is, you have to do biopsies and all that, and also you want to follow someone over time. A lot of the things are based on symptoms. So in your research, and I know maybe it's not applicable to all of you, but if you could tell me, how do you think your research can help clinical trials in the future? And maybe we'll start with Shalev.
So for example, when we want to see regeneration of the villi, of course, we're going to get the cells that we're losing are very specific cells that we know molecularly what they do. They absorb nutrients, they absorb lipids, amino acids, glucose. We know what are the genes involved. And so basically by measuring the transcriptomics in stool samples, we have all these genes that disappear in the stool samples of celiac patients, which are exactly this. The genes are expressed in the zones that are lost in celiac disease. And hopefully what we hope to see when we do these experiments, these measurements, is that upon a gluten-free diet or some successful drug, we're going to see these genes reappearing. And that will be sort of a non-invasive proxy that the tissue is healing, that there is mucosal healing. Of course, the alternative is to take a biopsy, but and there's it's a complementary approach, of course. Biopsies can morphologically show you that the tissue is recuperating, but molecularly measuring the cells is also, I think, a potentially powerful proxy for that.
So I guess from my research, I guess there are two things that are really relevant to clinical trials. So the first is when we do a clinical trial, it is a great opportunity to learn more about the disease as well as learning about the drug. And that's a really important thing. There are loads of insights that have come from recent clinical trials in celiac disease that have, as Jocelyn said, sort of inadvertent commas, failed, but they have taught us a huge amount about celiac disease. They have made for better clinical trials. And one aspect of that is to try and work out who will respond to a treatment and how we monitor that response. So, particularly when we go into phase 3 clinical trials, it is an opportunity to trial biomarkers or take samples where we can retrospectively look at potential biomarkers to say, hey, can we have a test that will show us whether this treatment is working in a particular individual? And that means that maybe we can then stop that treatment if it's not working earlier, save the patient money and time and risk if that treatment's not going to work for them. And maybe it can identify which patients are likely to respond to the treatment, so we can predict who a treatment is best for. So that's a real opportunity for sort of biomarker research in clinical trials. And it is a companion, because if you have a great treatment, but you have no idea which patients will benefit and which won't, that's not great. But if you have a good treatment and also have a good set of tools to predict who will respond and have a way of monitoring it, that is super helpful. The other thing that we do in our lab is this technique called spatial transcriptomics, and then these other kind of omics techniques where we look at gene expression in lots of complex ways. And one of the opportunities of clinical trials is to be able to understand more about the disease by using samples from a clinical trial where things are really tightly controlled. We can understand who the patients are, exactly what's happening to them, and things are collected in a standardized way. And that's a really sort of special set of data. And so those samples that patients have kindly donated by enrolling in clinical trials, those should be used to sort of maximize our understanding of the condition. So it's a great opportunity to apply some of these techniques to learn more about the disease as opposed to just learning about the drug.
Thank you.
So I totally agree with because I think it's two factors is quite important when do the clinical trials, and I think even other diseases also same. Can we identify the homogeneous group of disease? The celiac patient is really homogeneous or heterogeneous in terms of their symptoms is quite different. And their presentation is quite different. Some patients have micronutrients deficiency, some patients did not. And some patients actually develop lots of vomiting or lots of other symptoms. And even after treatment, after treat the celiac patient, they have present quite differently. And then is there any way to clustering those patients? I think some other autoimmune disease right now using about genetic variant and also using their clinical profiles, they try to cluster patient. So I think we should apply it in the celiac disease too. I have another study is planning to do is kind of using genetic component, I think exome sequencing data plus clinical profiles that including all laboratory findings and ferritin, some other micronutrients level, and hopefully we can cluster patient. Then we maybe select about the right treatment for the clustering, kind of applying to some clusters, and then we can have some homogeneous group to actually and try on the clinical trial. That's one aspect. And the second aspect is the outcome. How can we kind of measure outcome? This is really, really big, I think, the challenges. And also because the outcome measurement is expensive. You kind of maybe using about really kind of lots of expensive technology or advanced technology is really, really hard to. Even if we have found some drug or medication, the price could be really expensive because of some other many clinical trials used in identifying actual drugs. So is there any way to cheap price to measuring outcomes? can we just avoid endoscopy exam for kind of biopsy or historic challenges? I think we published one data and about 5 years ago using about peptide microarray that is measuring immune reactivity against gliadin and TTG based from, and like 12 moles from gliadin and 12 moles from TTG and even complex together. And we showed really, very promising data and can predicting actually the healing status when at the time of monitoring disease. And we have separate healed treated celiac patient, unhealed treated celiac patient, and TTG-IgA still has a negative non-healed treated patient, but our data showed the increased immune reactivity against some gliadin epitopes. We call it neoepitope or some epitopes. And those kind of very cheap methodology can measure outcome and will be, I think, the great for the helpful for the clinical trial, even reducing the drug and after actually discover or developed later on.
So, I don't know, Sónia, if you have a comment?
Yeah, so my research is at a preclinical stage. So, of course, the thing that the preclinical research can contribute is with leads, molecules or probiotics in the case, to start a clinical trial. So I think everything, so as Michael said in the previous question, everything is important and everything can contribute. And in fundamental research, we can have expected and unexpected results that can lead to a potential new clinical trial. So
I actually want to go to Sónia to ask you a question. You had mentioned about diet and the difference in what you're seeing in the microbiome depending on the diets, and that patients with celiac disease, I just think that the audience would be really interesting to hear your findings about like fiber and what you found would be very interesting to people.
Okay. Yeah, so I did, so the study was comparing the gut microbiota of celiac patients with their first-degree relatives. And so we saw that, first of all, we saw that so the kinship approach. So people that are related, family related, have a more close gut microbiota than comparing celiac patients with non-celiac. So that was one of our findings. And the other thing was, when we looked at the celiac disease gut microbiome, we realized, so we studied the gut microbiome and their dietary patterns. And so we realized that the gut microbiome of celiac patients had, I can say they had lower abundance of fiber foraging species. And so, of course, we only observed, it's an observational study, but when looking at the literature, those species that can degrade fiber are related to better health outcomes. So if a celiac patient has low abundance of these species, maybe that can, I don't know, explain part of some symptoms that they may have. And also, so as inflammation, etc. So that's one of our findings was exactly that. So, and the conclusion was also that it's important for the diet of a celiac patient to increase fiber consumption, so that they can also increase the fiber foraging species in their gut, because they have it, but in lower number. It's not that they're not present, they're just in lower abundance. Of course, we didn't study the exact impact that may have in patients, but we did see it, so it was comparable. So there was the fiber degrading reduction and also a lower fiber intake. And another interesting thing we also saw was regarding the nutrient supplements. There was some correlation between some species and the nutrient supplements, that celiac patients tend to take more nutritional supplements than the non-celiacs. So I think that's, I don't know if I correctly answered your question, but the diet may impact the fiber intake, and that may influence the gut microbiota, and that may lead to possibly higher inflammation status, etc. So that's that.
So this is a case, of course, where there's a finding. You have to go the next step, of course, to then prove whether or not the finding has an influence in health. So that's where the bench to bedside comes in, is that you investigate, you find something out, and then you have to prove it. So I think that's, where we're at, is we then now have to prove that.
Yeah, exactly.
Yeah, it's very interesting. So we've talked a lot about how your types of work can influence health. I'm just wondering, stepping back, do you see that your research could help prevent celiac disease from occurring in patients who have the genetic predisposition? Because we know that not everybody gets celiac disease who has the genetic background, and only some people do. So how do you see, and anyone can start, but how do you see your research maybe impacting that part of just the getting of celiac disease? Anybody?
Yeah, I think I can start again. So looking at this, the thing that I just mentioned, so if there are differences in the gut microbiome, of course, and you wrapped it up quite nicely. So first we observe it, and then we have to look and research further if it's the impact or the mechanisms or what, etc. But so for me, the thing that strikes me most when I think about celiac disease is it's exactly this. Why does a person that has everything to be celiac doesn't get the disease and the other does? For me, as a microbiologist, I think the microbiota can have a role in that, and so that's how my research can contribute. And if we could find the microbe that is associated with the triggering, so maybe a microbe can do two things or more, of course, but the microbe itself can have an epitope similar to that can be identified that by the antibody and trigger the disease, or the microbe can degrade the gliad into more immunogenic or less immunogenic epitopes, and that can lead to disease triggering or not. So that's that. I think that's a perspective that deserves being explored.
So for those of you who don't know what an epitope is, it's on the protein, it's the thing that triggers the immune response. So that's what she's talking about in terms of, gliadin being or gluten being digested. Some are going to be immunogenic or cause an immune response, and others may not be.
Yeah.
I wanted to add something.
Yes.
So in terms of my research, we've seen that shed cells from the gut that appear in the stool are sort of the normally shed cells, but when we analyzed endoscopic washes, basically the cells that are shed into the upper gastrointestinal tract, we feel that we're enriching for the cells that are actively killed, actively eliminated by the immune system. And I think that's an interesting angle because we know that the nutrient-absorbing cells along the walls of the villi are very heterogeneous. There's not one type of enterocyte. There are several subtypes. An enterocyte at the tip of the villus and at the bottom are as different as an enterocyte and a completely different cell type, like in mucus-secreting cells. So they're very, very heterogeneous. And I think one interesting open question is, are all enterocytes attacked equally by the immune system, or is there a particular molecular makeup that makes an enterocyte more amenable to attack? And I think by measuring the cells that are actually dying more together with the cells within the tissue, we might be able to find specific subpopulations of cells that tend to be attacked, and also to look at their in a spatial manner, like Michael mentioned. So in a spatial manner, once we find these cells in the tissue, we can see what are the types of cells in their microenvironment. Are there specific subset of immune cells that tend to be more violent? So that's one interesting angle that I think potentially we could shed light on.
Okay.
So, okay. The prevention is really important, I think. And also, I think there are many drug trials, working on trying to immune tolerogenics and kind of turn them back. And the recent our epidemiology study actually looking at why that abruptly increased celiac epidemiology in the world. And then our group we're looking at about whether it's actual more immunogenic gluten product and introduced our society. Therefore, they might have increased incidence or prevalence of celiac disease in the world maybe happen. And then we actually tried to using about, because of, right now it's a current industry, it's using about lots of viral gluten, which is kind of very triggering this celiac disease. And then we've tried to find out which gluten, what kind of methodology to cooking could be involved about more immunogenic bread, making more immunogenic bread. With something like that, those kind of study, it could be, preventing actual disease development. And also, right now I'm focusing about preclinical serum samples, which is meaning we really want to find out what kind of factors really involved. Actually, someone has developed celiac disease, autoimmunity. Someone has a positive celiac disease serology, but they're going to turn negative in the later life. So we really want to know about what factors involved, why those people actually having showed about transient fluctuation of the celiac disease immunity. If we found those things, we can maybe using about that findings to preventing disease development too. But this is really kind of really early step. We have to kind of go further that.
So it's not the main focus of my research, but it's not going to stop me sharing my thoughts. I guess the question for me is, why doesn't everyone who has HLA-DQ2 or DQ8, the genes that make you susceptible to celiac disease, why doesn't everybody get celiac disease? And we don't really have a good reason why 30% of the population don't have celiac disease. We know that there is this thing called oral tolerance. So animals have mechanisms to not develop a harmful immune response to stuff you eat. And then that is lost in conditions like celiac disease. And what we don't know is whether that is in celiac disease, whether that is a deterministic process, i.e. if X and Y happen, you will definitely get celiac disease, or whether it's a kind of a stochastic kind of random thing, whether you have a chance of developing celiac disease and certain events or risk factors shift the probabilities of someone flipping over into celiac disease or not. And whether they are dietary factors, whether they are microbial factors, viral infections, other environmental factors, other genes that we know play a role in the risk of getting celiac disease, we don't know which of those factors shifts the balance. What was mentioned about potential celiac disease is, I think, a really important thing, because we have, with people with celiac disease, this opportunity to study this precondition. So, why does somebody go from not having raised celiac antibodies to developing raised celiac antibodies? And why do they then go on to, or not go on to, develop intestinal damage? Now, I think the answer will not be found in a blood test, because I think probably what is going on is localized within the mucosa, within the lining of the bowel. And it is the interplay of these triggering factors and the immune cells and the epithelial cells that line the gut that cause this shift. And so what we need to do is kind of combine some of the epidemiological studies where we identify patients who are at risk of celiac disease, and then combine it with much more mechanistic work looking at the microbiota and looking at the interplay with the immune system and epithelial cells in the lining of the gut to understand those things. So there have been some really nice studies in animals looking at this, but it's a much harder nut to crack to do this in human studies.
And that's why we get all of you together, because you're all doing very different things that are complementary. So, Sónia, did you have something else you wanted to say? Or?
Yeah, no, just when Michael started talking, apart from all this, it's also intriguing why a kid at 1 year or a few months gets the disease immediately, and other people only when they are trying to get pregnant, because it's more prevalent in women. And so it's very intriguing and very interesting. So the age factor, everything.
Yeah.
I was just...
Yeah. So I want to give an opportunity for anyone in the audience to ask any questions. If you want to come up to ask a question, but in the meantime, I'm going to ask the panel. Is there anything you want to make sure that the audience knows about the work that you're doing and what you consider the really important prospects?
Can I share a random thought, which is an answer to your
Yes.
question?
Okay. Random thought. Yes.
No, it was actually just listening to the talks about epithelial cell alterations and the microbiota today. The epithelial cells are the ones that have all of the kind of absorbative and kind of catabolic machinery. So they are what help break down our food into its component parts. They alter it. And we know that the microbiota in the colon is fed by what we eat. But what actually gets to the colon depends on what's happened in the small bowel. So there is an interplay between not only what we eat, but what our enterocytes, what our small bowel does to it, which then has an impact on the microbiota.
Or the
So it's
opposite,
just
or the micro impact the food and then impact the
Yeah,
colon.
so there's a lot of interesting interactions. But I was thinking about some of these things that feed your colonic microbiota that we eat. Actually, whether they get to your colon or not depends on whether we have the machinery on epithelial cells to break it down.
I
So
mean,
it's just a connection
there's
between
many
these two.
more microbiota in our GI tract than there are
exactly.
cells of ourselves.
Yeah.
So, Claire, why don't you ask your question?
I'm asking this question for a friend.
Okay.
So in 2016, the mesentery was given its own classification as an organ, and I didn't know much about it until the last couple of weeks, but it's in close proximity to the entire GI tract. And my friend's question was really much more about what is the future for investigating the role of the mesentery in both the impact of the gut-brain connection for neurological or psychological symptoms in celiac disease. But in this conversation, it made me wonder about potential use of the mesentery and changes in it as biomarkers for disease expression. Anybody care about the mesentery?
Well, that's a very interesting question. I never thought about it, relating mesentery with the disease. Possibly there is something related. So it's a very recent organ. So I did my medical graduation in a few years ago, when I started studying, it wasn't even an organ, and in the end, then it was. So I was kind of relieved, because in the anatomy classes I had one less organ to study, and my colleagues afterwards had another one. But that's the thing I think about mesentery. Possibly it's related. The only thing I'd like to say, and also relating with the previous question, is was when I was hearing a question. So again, I'm going to talk about gut microbiota. So there are so many things that we still don't know, of course, and that's very interesting. But for instance, serotonin, which people relate to the brain, etc. It's much so the amount of serotonin produced in the gut is so much higher than the one produced in the brain. So we still have lots of things to know in the gut-brain axis. Maybe mesentery has something to do with it, but I never thought about it. I'm sorry, I didn't reply exactly to the question, but
No, that part of my asking the question was, hey, maybe somebody will start thinking about the mesentery.
Yes,
So it
exactly,
does have a
yeah.
role in immune system education and inflammatory response. And so it's like, oh yeah, that does kind of sound like celiac disease connection in there. So
Exactly, yeah.
thank you.
So there's a lot of interest in the mesentery and in inflammatory bowel disease, particularly in Crohn's disease, because there are a lot of not only immune cell structures, but also what are called stromal cells, which are involved in in kind of shaping tissue and creating the signals for how it kind of develops and regenerates. One of the problems that we have with celiac disease is that we study what we have access to. So we can access the mucosa, the inner lining, but the mesentery is kind of outside the supportive tissue, and bar somebody having a major type of operation called a Whipple's procedure, you can't get at the mesentery. So it is hard to study. That does not mean that it is not important. It just means that it's hard.
Yeah, and I didn't know if there were
it is
blood
a
tests
great,
that could...
great suggestion.
Yeah. So,
I also want to add that I think in every tissue, but specifically in the gut, all of the cell types are tightly connected with each other and communicate with each other. Neurons, immune cells, epithelial cells. Just one example is that, celiac disease is a disease where we lose the enterocytes, the cells in the outer lining, but the entire villus collapses. The villus has other components. It has fibroblasts, endothelial cells. Everything collapses. So sort of to keep the viability of these stromal cells, you have to have signals from the epithelium. So I think it's a very stimulating and interesting question. Definitely in irritable bowel syndrome, there's a lot of involvement of neuronal signals, neurons, and it remains to be checked whether it's celiac.
new frontiers.
Yeah, I mean, really, the GI tract is the largest immune function area, has its own nervous system, and it's very complex. We're learning more about mast cells and other cells that were really ignored for many years. So there's so much more research that needs to be done.
As I mentioned, it's also the largest endocrine organ in our body.
Yeah.
The most hormone-producing cells are, even though this is only 1% of the cells of the lining of the gut. And again, these are not attacked in celiac disease, but they live among the enterocytes, and so we might be losing them. So there's a lot of other signals that could be involved.
Thank you.
I think that's a really amazing idea. I agree, because we have mouse model. When you do study the mouse model, I think mainly kind of taking out mesenteric lymph node. We haven't thought about, I don't know, any literature and already published using other mesenteric lymph node for the celiac disease. But I think that's a really great idea. Maybe based on some surgical sample specimen available, then I may need to kind of look at about what kind of immune function has happened at mesenteric lymph node in humans. I mean, the same as the mouse model we did.
Mm-hmm. Okay, another audience question?
I am Catherine Johnson, and I'm not affiliated with anything except I have celiac disease. I have questions for all of you, but I'm not going to ask all of them. Dr. Pereira, when you talked about I'm thinking chicken and egg with the fiber, and if there's less fiber attacking microbiome, is it because people are eating less fiber, some of which is sort of forced on us, or is that a thing that's inherent to our bodies and the way we've responded? But I'm wondering, that's certainly an avenue to, okay, you found a thing, now let's go research that. How do you improve the fiber outlook for someone who has celiac disease and needs to stay on a gluten-free diet, and does that change that? And I would love to see the results of that. The other thing is, you mentioned when you did the family trials, and you were comparing people with celiac and people without celiac in the same family, and one was taking more supplements than the other. Did you do any follow-up to see whether the people who were taking the more supplements, with the people with celiac who were taking more supplements, had a different panel of, for instance, I take a ton of supplements, and my doctor and I fight about this all the time. You're taking too much of vitamin C, you're taking too much of vitamin E. So I said to him, okay, run it. Tell me how much I have that I'm able to use. And he said, take what you're taking, after he did those studies, because I needed every bit of it. So, did you do follow-up to see all the people who were taking the more supplements? Was that because they needed it? And was that actually what they needed in order to support their systems?
Yes, I would love to say yes, but unfortunately no. So it was an observational study. One observations were cross-sectional. So we had the questionnaire and we had the sample, and then we analyze it afterwards. So that's what was possible to do. It would be a clinical trial to do a follow-up, etc. It would be very interesting. So the thing is, we only collected information, so we didn't screen if people needed the supplements or not. We just related, so those that are on nutritional supplements and these aren't on nutritional supplements. Regarding the fiber intake, so I pointed out also in the article, so we are indeed comparing people. Of course, they are family related, and that reduced variability, but they are still on a different diet. So the celiac patients were on a gluten-free diet.
Right.
So again, you started the question with the conundrum. So it's the egg or the chicken. So we don't know. It's an observational. So what we saw is that there is a low fiber in their diet and low fermenters of fiber in their diet. And then we speculating, not the correct word, I don't remember a different one, but then we looked at the literature and said, okay, so these bacterial species that are decreased, that degrade fiber, they are related to less inflammation, etc. So that's the only thing we could look at. We weren't able to check the inflammatory status of the participants or the nutritional supplements they were having. Unfortunately, we couldn't do that. Sorry.
Okay. Well, that sounds like a great follow-up study, doesn't it? And one of those things that you were speaking about, that we fall over things. I have another question from the morning. When you were talking about doing the fecal assessment before a colonoscopy and the information you were getting from that, was that after someone had done normal colonoscopy prep and you could still get enough information from what was left?
Yeah, so our initial studies were done on fecal samples obtained just before colonoscopy, after the 2 days preparation that cleaned the gut from bacteria. And there we simply, of course, most of the molecules originate from bacteria. So the fact that there were less bacteria enabled us to catch some of the human transcripts.
So you were still able to do that even though people had done their full prep?
But of course, this is not scalable, and we wanted to do this also for celiac disease and just, sample individuals at home. So there, for that, we developed this genomic depletion protocol for bacteria, where we basically where we can handle the large amount of microbes in the stool, and we basically deplete the majority of them. There's about 14 species of bacteria that take up about 90-something% of the bacteria in the gut, and 90% of their RNA molecules are ribosomal RNA. And so there's actually a small set of molecules that if you can specifically degrade, you get rid of many bacterial molecules that are masking the human signal, and that increases the amount of RNA molecules from shed cells by about 6 to 7-fold. And that's enough to actually get a rich signal from stool samples.
That's a very exciting development.
Thanks.
Now, all of you who are presenting, there are lots of slides that have gone by, and I was thinking that was what we were going to get when we checked the QR code, and I'm not seeing that. But a lot of what you all put up there was so interesting, but far, far away and very little. So I'm hoping that we can get access to more of that material.
Yes, so all of what was presented today will be on our site as the whole conference. Amy or Kate, I don't know where Kate is.
I'm hoping we get the slides, not just a video, which has the slides as far away as they were today.
So we'll talk about whether or not we can do that.
So I know that's your material, but a lot of it was really interesting, and I'd really love to look into it more.
Okay,
Thank you all. This has been amazing. Thank you.
And I think we have one more question from.
Hi, I'm mom of a 9-year-old boy who was diagnosed with celiac at the end of June this year. And he's always had a high ANA, not rheumatic related. What's your experience, if you have, with a high ANA and celiac?
Fitz from a.
I'll start with I'm not a pediatrician, so I may not be the best person to say this. A whole range of autoimmune conditions have kind of shared risk factors. So there is a whole panoply of genes that slightly increase your risk of a range of autoimmune disorders. So having one autoimmune disorder can put you at risk of having autoantibodies against other autoimmune disorders, and can increase your risk of having those other autoimmune disorders. But sometimes people will just have a raised ANA or a raised antibody are related to another autoimmune disorder, a little bit like potential celiac disease, but not go on and develop any other related autoimmune disorder. But it is important when evaluating somebody with celiac disease to check carefully that you are not missing somebody who actually has two autoimmune diseases at the same time. So that's something that we would sort of routinely check for in adult celiac practice. But as I said, I'm not a pediatrician.
Yeah, I was just curious as to know if your patients, and if any of them had a high ANA.
Yes. So sometimes we see people with a detectable ANA, and sometimes that's not related to any other disorder.
Correct.
And sometimes it can be because they have a second autoimmune condition. And again, we don't really understand that much about why these are related, and why sometimes we'll see an adult who has not had any autoimmune problems for many years, and often it's a young woman after let's say a pregnancy, and then they will go on and develop 2 or 3 autoimmune conditions in a relatively short amount of time. So sometimes a trigger, as we were talking about triggers of autoimmunity, it may be that some of these triggers are shared with other autoimmune diseases, and that developing celiac disease can then end up being associated with the development of, let's say, thyroid disease or type 1 diabetes or so on, and they can kind of coexist.
And then my other question is, do you find that the ANA goes down once the celiac is under control?
I don't think I've looked at that thoughtfully enough, but you're nodding. So I don't think I've seen any data about that.
Because autoimmune disease and celiac disease, because I'm not going to give you medical advice. Okay.
Not looking for it.
So autoimmune disease and celiac disease, many actually overlapped about NSLE, rheumatoid arthritis, and skin diseases, AA, and vitiligo. There are many autoimmune diseases that overlap. Sjogren's syndrome, about 5 to 10%, sometimes a little higher based on the population. And also based on interesting kind of finding is someone has a gluten-free diet, and then they're gonna go, even their autoimmune is also kind of, having well treated. And type 1 diabetics also kind of really hard to treat, because even with celiac disease and having some problems, but gluten-free diet and sometimes really kind of good benefits and for treating autoimmune disease. I think Jocelyn has much more answer.
Thank you very much. So I want to thank the panel, and we're going to take a 10-minute break. So be back here at 3:15.